Keep in mind, all information here is being researched, comprehended, and delivered by a 16-year-old high schooler. Take everything in this video with as many grains of salt as you can fit in your greedy little mouth. Feel free to return the salt to its natural habitat through the comment section. It's always a good idea to do your own research, make your own conclusions, and think for yourself. Hi, I'm Adrian from Earth, and today I want to talk about the Ice Age. Now, the movies were great. All 7 of them. 7? Really? Huh. Seems like a bit much to me, but okay. But what exactly was the Ice Age? The Ice Age is the common name for the Pleistocene epoch, which, towards the end, is when the last glacial period happened on Earth. Or, in other words, the Earth got a little chilly and its mother made it put on a jacket. This glacial period lasted from about 110,000 years to 11,000-ish years ago. We know at least a little bit of what the world looked like during this time, especially the animals. We have fossils of those animals, so the evidence for their existence and abundance can be clearly seen. We all know about the mammoths and rhinoceroses of the woolly variety, and the walrus cats and the sheep birds. There was even an early human walking around called, quite seriously, the Homo erectus. But what if you were asked what the plants of the Ice Age were? What types were most abundant? What did they look like? In your effort to guess, you might say the same sort of vegetation that exists in cold places today, like Canada or my heart. You might say grass, pine trees, and woody bushes. Science would agree with you to an extent. But before we get into that, let me say one thing. Animals during the Ice Age, as they do now, bleed, shed, poop, die, all sorts of things. And this gets trapped in frozen water. Frozen water? What are you, big ol' idiot? That's called ice! Oh. These things that animals leave behind, like their sheddings, their blood, their poop, their dead bodies, can get trapped in ice and permafrost. Now, plants, they're cool. A lot of them make pollen, and this pollen just gets everywhere. And this includes ice and permafrost. And plants also die and, get their dead juices all over. Using this, scientists were able to conclude which plants were more abundant during the time. Some of them used pollen data to conclude that grass was the most abundant, but other scientists, using different data, reached a different conclusion. This different data has to do with DNA. This process they used to gather data is called DNA barcoding. DNA barcoding has many uses and applications, like identifying new species, figuring out what animals have been in an area, or even attempting to put the DNA sequences of every animal on the planet in one database. In this study, headed by Eski Willerslev, DNA barcoding was used to identify the abundance of plants and the plant types in the Ice Age. This is important to these scientists because understanding what the plants were like in the past can help us understand what happened either to those plants or as a result of those plants, and can help us predict and plan for the future. Now, DNA barcoding has a general procedure called the DNA barcoding production pipeline. First, you have to have something to test, whether that's a dead animal or uprooted plant, or in this case, a flippin' lot of ice and permafrost. Then you extract the DNA from that thing you have chosen through a long, long process of taking a tiny sample, putting it in a tube, mashing it up, pouring in some science-y liquids, centrifuging and incubating, and centrifuging and incubating, and centrifuging and incubating, etc. Now, after you've extracted the DNA, you have to amplify it so you can sequence the DNA. You do this using a PCR machine, also known as a thermal cycler. The PCR technique is really interesting, so let's talk a little bit more about that. The credit for the PCR technique, which stands for polymerase chain reaction, goes to a man named Kary Mullis in 1983. I just want to mention real quick that Kary Mullis, after creating the PCR technique, said a lot of things, some of which are in his autobiography entitled Dancing Naked in the Mind Field. For example, he joyfully announces to the world that he was on LSD just all the time, and even that LSD helped him create the PCR technique. He also said he doesn't think HIV causes AIDS, or that climate change even exists. And to top it all off, he claims he saw a glowing raccoon one time, and no, LSD was not involved. Anyway, PCR. It uses a lot of heat cycling, hence the machine name, and enzymes to replicate the DNA a frickin' bunch of times. During the PCR process is when you would add the primer for the gene that tells you what your sample is. If you're familiar with the process of DNA replication, this is how the DNA polymerase attaches to the DNA strand and creates the complementary strand. In most animals, you use the CO1 gene, which isolates the gene that will tell you what your species is. But in plants, you have to use a different one, the MATK or RBCL genes. After the DNA is amplified, you sequence the DNA, and the scientific community basically holds a tribal council, and Jeff Probst banishes your data if it doesn't meet barcoding standards. Assuming you don't get your torch snuffed by Mr. Probst, you then use and share your data with the world. The researchers in this study used the DNA barcoding process by taking over 200 samples of permafrost and ice and identifying the DNA in each one. And they also took samples from the guts of dead Ice Age animals and identified the DNA that they'd been eating. Using this, they concluded that grass was not the most dominant plant during the Ice Age, but actually nutrient-rich clovers. And they even postulated that this might be why the animals during the Ice Age got so strong and big, is because these clovers were so full of nutrients. So, in conclusion, this study was important to us in our everyday lives because we now know where the first Lucky Charms came